In this video, we will be diving into a fascinating topic and one that is crucial to understand in the field of medicine, the tetanus toxin. This toxin is produced by the bacterium Clostridium tetani, and this powerful neurotoxin can lead to severe muscle spasms and can be even life-threatening. But how does it work, and what can we do to protect ourselves? Let's get right into it. Let's take a look at the structure of this toxin. It consists of two polypeptide chains, a heavy chain responsible for binding to nerve cell receptors, and a light chain that interferes with neurotransmitter release. These chains are connected by a disulfide bond, and the toxin appears as a small complex protein that can act at extremely low concentrations. Let's now look at the modes of entry of this toxin into the body. One common route this toxin can enter the body would be through deep puncture wounds, such as when a person steps on a rusty nail, particularly in gardens or construction sites. This can introduce C. tetani spores. These spores are highly resilient and can survive in harsh conditions, including extreme temperatures and lack of oxygen. Animal bites from dogs or cats can also lead to contaminated wounds and increase the risk of tetanus if not treated earlier. Traumatic injuries like fractures or deep cuts can also pave the way for Clostridium tetani spores to enter the body. Next, post-surgical infections could occur if the surgical site is not adequately cared for. Another common way would be when humans can get grass awns lodged in their skin while hiking or gardening. And lastly, some individuals could experience cryptogenic tetanus, which means they could have developed the disease without a clear history of the wound. Now, you've seen the possible ways this toxin could enter the body. What happens next? Well, let's take a look at where this toxin goes once the body has had exposure to it. After germination of the wound site, the bacteria Clostridium tetani multiplies and produces a tetanospasm toxin in an anaerobic environment, or without oxygen. This begins to disrupt normal nerve function. This multiplied growth is often associated with deep puncture wounds where oxygen levels are low, creating a conducive environment for the bacteria. As the bacteria proliferate, they can cause local tissue damage as C. tetani secretes various enzymes such as proteases and phospholipases that can break down the cellular components. This damage can lead to increased permeability of the cell membranes in the surrounding area and produce an inflammatory response. Inflammation causes the release of mediators that can alter the permeability of endothelial cells, or the cells lining the blood vessels. Also during this time, the bacteria are germinating in the wound, and this is when they release the tetanospasmin toxin. Due to increased damage and increased permeability allows molecules, including the tetanospasm toxin, to leak out of damaged cells into the extracellular space. This disrupted cellular environment can create a favorable condition for the diffusion of the tetanospasmin toxin from the interior of compromised neurons and their cells into extracellular fluid. As the concentration of the toxin increases in the extracellular fluid due to local production and release, a gradient is established. This gradient can drive the diffusion of the toxin into the surrounding tissue and eventually into the bloodstream. Once in the extracellular fluid, the toxin can be absorbed by nearby capillaries or larger blood vessels.
After the tetanus toxin enters the bloodstream, it binds to receptors present on sensory nerve terminals. By binding to these receptors, the tetanus toxin is able to enter the nerves of the peripheral nervous system. The specificity to enter PNS sensory neurons over others is due to their proximity to the site of infection. The nitrogen coating around the nerve cells attracts the tetanus toxin to them. To gain entry into the neurons, the tetanus toxin interacts with the ganglioside receptors on the surface of neurons. These gangliosides are believed to be associated with the tetanus toxin receptor, which, when bound, triggers uptake into the neuron. Once internalized, the tetanus toxin is transported from the nerve termini to the cell bodies and the central nervous system. Here, the toxin is able to reach inhibitory neuron terminals. In a normal neuron, glycine and GABA release and act on motor neurons to block excitation and the release of acetylcholine, which induces muscle relaxation. Let's look at this schematic. First, we have the site of infection, the skin. Then we have the PNS neurons synapsing onto the interneurons in the spinal cord in black. Then The interneurons lead to the motor neurons, and the motor neurons, the muscle. We'll keep this in mind as we go through. But first, let's take a look at how glycine and GABA act in normal conditions. Glycine and GABA act as inhibitory neurotransmitters in the CNS. GABA and glycine open ligand-gated anion channels with receptors specific to them. Upon activation, GABA and glycine receptors permit the permeation of chloride through these ligand-gated anion channels. The influx of negatively charged chloride inhibits postsynaptic cells. In this case, that is the postsynaptic motor neurons. The channels that you see where glycine and GABA bind respectively to trigger the release of chloride across the membrane into the postsynaptic cell. Therefore, they act as inhibitory neurotransmitters and prevent an action potential from firing down the motor neuron and thus induce relaxation of the muscle. However, when the tetanus toxin is introduced to the bloodstream, GABA and glycine are affected. The tetanus toxin only affects skeletal muscle, while smooth and cardiac muscle remain unaffected by the toxin. This is an indirect effect on skeletal muscle. Let's pick up where we left off. The tetanus toxin is using PNS neurons for transport to the spinal cord. Here, we are seeing the PNS neurons in blue synapsing onto the interneurons in the spinal cord. This is synapse 1. We will zoom in here. Once in the spinal cord, the tetanus toxin has a choice. Which neuron should it spread to next? Before the target can reach its end goal, it encounters excitatory and inhibitory interneurons in the spinal cord. We know its preference for inhibitory interneurons, GABA and glycine, but how does it only target them? As the tetanus toxin searches for the next neuronal target in the spinal cord, it targets a specific receptor. Scientist Ye and his team found that this receptor is SV2. But we run into another problem. Both excitatory and inhibitory interneurons in the spinal cord have SV2 receptors. However, the effect on excitatory interneurons is negligible because the tetanus toxin prefers inhibitory interneurons. So how is this preference established? The answer lies in the two orthologs of SV2. It turns out SV2A is concentrated to the GABA and glycine inhibitory interneurons, while SV2B concentrated to the excitatory interneurons. It was found that the tetanus toxin has a higher affinity for SV2A and therefore GABA and glycine inhibitory interneurons. This is how the tetanus toxin targets inhibitory interneurons over excitatory interneurons. Once the tetanus toxin targets the SV2A receptor, it binds and enters through a Trojan horse strategy. In a normal presynaptic cell, the vesicles release neurotransmitters and intermingle with the membrane before they reform and recycle. During the point of intermingling, the SV2A receptor is exposed to the synaptic junction and the tetanus toxin binds. Then, when the synaptic vesicles are recycled, the tetanus toxin is able to enter the inhibitory interneurons. Once in the inhibitory interneurons, the vesicles travel from the termini to cell bodies where they synapse onto motor neurons. Let's go back to our schematic. So we've gone from blue PNS neurons to the black inhibitory interneurons. Now we're at the point where the black is synapsing onto the red. The red is the motor neurons. This is site 2. We'll zoom in here. So the tetanus toxin has reached the cell bodies of the inhibitory interneurons. Here, the tetanus toxin prevents the presynaptic release of GABA and glycine onto motor neurons by cleaving VAMP, aka synaptobrevin. First, the tetanus toxin releases its light chain. The light chain diffuses through the vesicle membrane and attaches to VAMP. Upon contact with VAMP, the light chain uses its zinc peptidase to cut VAMP and render it useless. VAMP is crucial for the release of GABA and glycine for synaptic vesicles. Without VAMP, the vesicles cannot fuse to the cell membrane and release GABA and glycine neurotransmitters. With the tetanus toxin blocking the release of inhibitory neurotransmitters, GABA and glycine, the motor neurons no longer receive inhibitory signaling. Since inhibitory signaling can no longer occur thanks to the tetanus toxin, somatic motor neurons become hyperactive from only receiving excitatory input, which leads to excessive firing. Lucky for the victim, the tetanus toxin does not enter autonomic motor neurons or visceral motor neurons. But why? Through its targeting of GABA and glycine inhibitory interneurons and higher affinity for the SV2A ortholog, the tetanus toxin only affects somatic motor neurons. This is because GABA and glycine interneurons only directly synapse onto somatic motor neurons. If this specificity did not exist, the tetanus toxin would be able to spread to all muscle types and the victim would perish. So, as the tetanus toxin spreads, it searches for SV2A receptors and binds, preferentially targeting GABA and glycine inhibitory interneurons in the spinal cord, which is why the tetanus toxin only affects somatic motor neurons and, as an indirect result, the victim's skeletal muscles. Therefore, it is through an indirect effect that causes the tetanus toxin to target somatic motor neurons. This is why we see a rigid paralysis instead of par- instead of a flaccid paralysis. The rigid paralysis induced along with the spasms characteristic of tetanus. But again, it has no effects on the heart or other organs.
As we mentioned, the lack of inhibitory neurotransmitter release, specifically GABA and glycine, lead to hyperactive motor neurons. But why? Tetanus toxin specifically blocks the release of inhibitory neurotransmitters from inhibitory interneurons, but it does not impede the release of excitatory neurotransmitters from excitatory interneurons. In tetanus, only these excitatory signals reach motor neurons, causing them to continuously receive signals to fire off action potentials to skeletal muscles, causing contractions. The motor neurons do not receive glycine inhibition signaling from the impaired inhibitory interneurons, so the firing of motor neurons is never inhibited, and skeletal muscles are continuously stimulated. The lack of inhibitory signaling from these interneurons leads to overstimulation of the skeletal muscles and tetany. According to Cleveland Clinic, tetany is a series of involuntary muscle contractions from prolonged overstimulation of motor neurons. The involuntary control and lack of recovery due to suppressed inhibition leads to eventual spastic paralysis of the muscle. Unlike flaccid paralysis, where the muscles are not receiving signals to contract, spastic paralysis is marked by stiff and rigid muscles that spasm occasionally involuntarily. From analysis done by Penn State, tetanus is typically known as lockjaw due to the characteristic spasms and locking of the jaw one experiences when inflicted with the tetanus toxin, but it has the ability to affect any skeletal muscle in the body due to how it spreads throughout the nervous system. Skeletal muscles include a whole slew of muscles, from muscles in the face, abdominal muscles, muscles in the limb, and muscles in the neck and back. When inflicted with the tetanus toxin, all of these muscles have the possibility of experiencing overstimulation and paralysis, depending on what inhibitory interneurons are affected by the toxin, what motor neurons would normally receive signals from these interneurons, and what skeletal muscles receive signals from those motor neurons. When the affected interneurons are ones that signal to motor neurons that control skeletal muscles found in the back, it leads to a condition known as epistotonus. Epistotonus, as defined by Stat Pearl's article on epistotonus, is characterized by a severe backward arched posture due to the overstimulation of skeletal muscles in the back, neck, and lower extremities. Continuous uninhibited contractions of these muscles lead to this contorted posture. The below painting by Sir Charles Bell illustrates this posture while additionally highlighting other uncontrolled contractions the tetanus toxin promotes by inhibiting GABA and glycine release in the inhibitory interneurons in the signaling pathway of the affected muscles. Note the contracted arms, hands balled into fists, the clenched jaw, and contracted toes. These episodes of intense involuntary contractions can cause tears in the muscles, muscle weakness, and fractures due to sudden contortions and overworking the muscles. While the overstimulation of skeletal muscles that cause epistotonus have one of the most striking visual effects on the body, overstimulation of other skeletal muscles have further detrimental effects that are not as visible as epistotonus. The muscles involved in swallowing are skeletal muscles, making the entire process of eating and drinking difficult, as an individual suffering from the tetanus toxin will encounter two major difficulties. First, the characteristic clenched jaw makes it difficult to get food into the mouth. Second, swallowing is a voluntary action, but those afflicted with the tetanus toxin may lose control of the neck muscles involved in swallowing, leading to swallowing difficulties and potentially choking. The chest muscles and intercostal muscles are muscles involved in breathing. While breathing occurs passively, it can be voluntarily controlled, and the contraction and expansion of these skeletal chest muscles allow for the lungs to contract and expand. When the contraction and expansion of these muscles are disrupted, such as when the motor neurons that control these muscles have been affected by the tetanus toxin blocking inhibition, the individual may experience difficulties in breathing and may have to rely on respiration machines until the toxin has run its course and degraded.
The recovery of the synapse is very slow and requires the formation of new nerve terminals and presynaptic structures, with tetanus toxin having half-life of about 5 to 6 days, meaning that it roughly takes 5 to 6 days for the toxin to become half of the concentration from the initial amount. This toxin can be broken down into the gastrointestinal tract due to the lack of a protective protein complex. This toxin can be broken down into 3 fragments: fragment A, B, and C. Fragment C is a non-toxic fragment that possesses an immunological feature that helps neutralize the lethality of tetanus toxin and is thought to be a key candidate for the vaccine against tetanus. The light chain, also named fragment A, has a zinc-dependent proteolytic activity responsible for synaptobrevian cleavage. The heavy chain is composed of the fragments B and C. Fragment B is located at the N-terminus of the heavy chain and is responsible for toxin cell penetration, while the toxin protein is completed by fragment C, located at the C-terminus of the heavy chain and is involved with neuronal cell binding. Another way that the synapse recovers is through toxin degradation, where multiple variations can occur, but in this situation, the most likely mechanism that would occur is microbial absorption. This is a surface phenomenon where heavy metals are positioned on the cell surface, and through absorption, the inorganic materials are absorbed into the cell. This mechanism consists of microbes collecting inorganic materials by attaching substrates to them, and then absorbing them through the gastrointestinal tract. Once the absorption has occurred and all of the inorganic materials are gone, it will stop the blocking of release of neurotransmitter at the inhibitory synapse, allowing neurons to once again send signals to other neurons and or tissues. A low pH in the stomach prevents microbial colonization with a pH typically less than 4, altering the chemical form of the toxin, making it more or less readily available to cross the microbial cell membrane. Generally, acidic conditions increase the absorption of acidic toxins, while alkaline conditions enhance the absorption of basic toxins, thus impacting the overall toxicity of the substance to the microbe.